Good morning, everyone. Today is our second Q2 presentation for the HPP BD initiative. It stands for Disease Proteomics. And it is my pleasure to have three representatives today of initiatives. First, I would like to invite a talk of Susanna Cristobal. She will present the IHOP initiative for us today. And I would like all the participants who is listening for the talk, please put your questions into the chat. And then in the end of each talk, I will read them for representatives, and then we can have a QA discussions in the end of the presentation. Okay, thank you. And Susanna, please start your talk. Thank you.
Hello, thank you for inviting us. Good morning, good afternoon. This is Susanna Cristobal from Linköping University and one of the chairs of the IMOP. So let's start talking about models in biology. We all of us, we have our favorite model to answer our question. And the reason is because obviously models play a crucial role in biology because they help us to understand, to predict, to explain this complex biology system. And all of these things come up to the classical key issues in models that are how we can simplify this complexity, but still using for testing our hypotheses, but still sharing this capability of reproducibility and also to elaborate further works. And we start discussing those key concepts when we decide to elaborate this initiative. And by doing that, we start publishing a publication in Communication Biology that we try to go into that. So in this era of genomics, of course, traditional models somehow are not large and sufficient for our problems. And I try to illustrate that putting this kind of Venn diagram of the concept of One Health when we integrate animal health, human health, ecosystem condition. So the question that arises, how should model organism, what should offer to our proteomics community? And we think that actually could help us for three different concepts that still were there gaps, and gaps including how we study molecular mechanisms of human health and disease, and questions that are not clearly answered, or also how better understanding pathology, pathology reservoir, medical assistant. But moreover, we need also to understand how toxicity affects us, how our exposome, which are the biological system, which are the specific sentinels that maybe help us to understand all of that. And this is what comes to the vision of this initiative and how we think that rethink the concept of model organism could help us to move from traditional organism to more diverse opportunity for organism. And that means that we include holobionts, we include sentinels, we include all diverse of organisms that could be more adequate for different questions, or also how to capture this biology complexity through diverse models that go into the concept of the tree of life. And we think that this working with this new perspective actually could help us to improve this, both aspects of human health, both environmental quality, but also biodiversity conservation. And from this perspective, in this initiative, we have defined a kind of objectives that we state. And I put it here, this text, but I think it's important that I mention a little bit that this includes promote the use of diverse, relevant models that could tackle unresolved biological questions. And to do that, we need to promote most advanced tools and techniques to put our knowledge of those models. And this means obviously including the concept of developed informative resources that could be improve our comparison between species, between models. Of course, also proteogenomics, multi-layer approaches that could improve genome annotation, identification of functional proteomes, non-canonical proteins, but also instigate the use of signal cell proteomics into valuable models to help to understand this heterogeneity. So the question is a bit why IMOD should matter for QPO, and this is why I'm here. And we think that this promoting of comparative genomics, proteogenomics, ecotoxicoproteomics in IMOP actually could support some of these gaps that we want to answer. And those include molecular fundamental mechanisms that still are difficult to study in humans and human systems, and also aspect of identify novel coding sequences or characterization of function of encoded proteins. There are also opportunities in that line. And as well into the aspect of how we monitor the environment in the terms of pollution, in the terms of fate of pathogens or impact of its quality in living organisms. Those are some of the directions that we think get into the umbrella of this initiative. And in order to get a little bit of illustration on that, I have selected a few slides with specific examples that give an overview of these ideas. So let's start talking about halobionts. And halobionts are a new type of model organism under our perspective, because include the host and the associated microbiomes. And this is often more inclusive and ecologically more valid for biology. So here the terms halobiont is not just bacteria, it includes archaea, includes fungi, includes virus, includes alga, protozoa, so reflect the deep interdependence and co-evolution of host of microbiota. So for truly understand these systems, of course, we need to go into more complex approaches, and certainly metaproteomics ideal and deep metaproteomics analysis nowadays can provide this kind of functional insight into these proteins that are expressed in each of the members of the microbial community, and how these pathways and this ecological role could be understood. So I have selected this picture from Jan Armengau group, and you can see here how this analysis of the holobionts, we can go beyond the type of information that the classical metagenomes could offer, of course, because we get quantification of taxa biomass, and how the identification of proteins and pathway, and how we can dissect and integrate the microbiotic functionality that is truly integrated with the host functionality. And this is the beauty on this particular opportunities. Obviously, we are not we are clearly aware of the difficulties, and the difficulties of reproduction. For example, holobionts models are certainly a challenge, but still is in the community collaboration, maybe standardization resources, we can have the opportunity to refine these complex systems. Let's go to different questions also, like if we think about how to tackle a question related with alternative proteins, and what we can think about host proteins, maybe as analogy to model organism for the different protein functions. How to investigate and refer proteins. Alternative proteins, those proteins that are not canonical reading frames, represent still something underlooked, but with a functional part of the proteome. And these molecules, and these represent a big challenge in the context of annotation pipelines and of these new questions. So, of course, we need new workflows that include deep transcriptome mining, ribosome profiling, and validation, and IMOP is part of this support systematic discovery of all proteins with the integration of those workflows. So we can still see that there are early findings that suggest that many ALK proteins, such as regulators, are involved in understanding human disease as well. So their function will actually reshape our understanding of gene expression, of protein complexity in all these species. More. Yeah, comparative evolution proteomics is also something that could help us to tackle biological question: how diverse organisms naturally adapt to environmental stressors. This evolutionary approach actually helps to identify mechanism of resilience, or guide the strategy in One Health, conservation, in innovation as well. So of course, we are talking about non-model organism in some way, and proteogenomics offers us today a full potential for study this organism. But of course, need to go into the utilization of new tools that are able to work with cross species to make it easy to generate high quality proteomics data in those emerging models. But when we put this together, we can also use it for investigate those questions in a comparative multi-omics approach, integrated proteomics with metabolomics, with other omics that enrich the opportunity to understand this mechanism, but across different life forms. And I will maybe conclude this set of examples talking a little bit about now human and also any species in the environment are constantly exposed. I mean, we are chronically exposed to very low concentration of a mixture of pollutants. And this is what we call today the exposome. And there's an awareness in the society about what this could happen, how this could involve also our impact and the impact in general. And we can get these advanced tools based in, for example, thermal protein profiling is an ideal tool to identify those proteins that are direct target of pollutant and mixture of pollutants, and get back the opportunity to get the molecular initiation event that is key to understand and key to start predicting what could be effects. And if we combine this with toxicoproteomics, as we mentioned also before, using proteogenomics with many different organisms, we can get the cellular response to toxicant across species. So by utilizing these metaproteomics methodologies, we can tackle both molecular initiation event and key events, and go into predictability of the impact, predictability of the possible risk for humans and for animals and for the environment. And this is a very short, let's say, introduction of our initiative, and try to get a flavor of what we are talking about when we try to present this model organism for proteomics. And I have put it together here, a kind of overall deliveries of our activities, and we start working together sometime in 2023, when we start brainstorming again about the concept of model organism proteomics. And we start by publishing this paper, diversifying the concept of model organism at the age of omics. And then we have continued also a little bit gathering this community, and we offer a kind of web seminar about new models and new proteins. We have continued our activities, and in HUPO last year in Dresden also, we also present one session of the community that attracted quite a bit of attention. Afterwards, we also published another paper this year about the novel model organism and proteomics as kind of better way of understanding biology. In the same line, we are going to have it right now in a kind of 1 week or 2 weeks in HUPA, in St. Malo, also another session of the IMOP community. And of course, we would like to increase the critical mass between you people listening to us and be able to plan and organize more seminars, more winter school or summer school, or also plan to work together in develop of new bioinformatics tools that could allow us to annotate human genome, perform biological interpretation of data, but also gain structural insight in maybe not canonical proteins, proteoforms. In addition to that, also we are working with Journal of Proteomics, and we are planning several specific special issues, and they're going to focus in this our community and this kind of gaps that we can solve with this initiative and these kind of objectives. And by doing that, I want to conclude, but before that, of course, I ask you to check in this information that you can have in HUPO, and in particular, my question is that we proteomic researchers, we have our topics, our specific lines of research, but we really can join more than one initiative because the initiatives are not really restricted. We can have objectives that also go into one and another. So please have a look, because this is open to all proteomic researchers working with animals, plants, environmental species, microbial species, holobionts, and human, and how we address this question together from the perspective of the model aspect that could help to our proteomics strategies. And with that, thank you for listening, and I'm happy to take your questions.
Okay, thank you very much, Susanna. It was a really nice talk. For me as well, I for the first time seen the presentation from your initiative, and it was very nice to hear and see what you do. I think the model organism are a very important part of the story, and this part of the story could be related to both area where we could just try to learn how the nature works on different type of species and then apply back to the human health, as well as, of course, in generally analysis of ecosystem of the planet, which is another very important area of the research, health, but now the planet health. I think these two together plays a very important and crucial role. At this point, I don't see any questions in the chat. That's why I think at this point we don't have any question to you. But again, thank you very much for your presentation. And I would like to mention that initiative did create a report for this year. And this report from this initiative and other initiative from the biology of disease will be published in the special GPR journal later this year. And so please welcome to look at this journal where all the significant publications will be highlighted from each initiative by the separate initiative. Okay, again, thank you very much, Susan, for your presentation. It's really nice to hear this initiative and see what you try to put as a community. Our next speaker today will be Benoit Conat. He will represent his initiative, Metaproteomics, and I would like to pay attention to this initiative because, in my opinion, I think this is very much past due initiative. It should be organized much earlier than it is, but we welcome you now as a new initiative and looking forward to see what you've been able to do so far. Please do.
Thanks a lot. So, yep, I'm Ben Conat from the Luxembourg Institute of Health, and I will introduce this Metaproteomics initiative. I don't think we've done it already in such format, at least I didn't do it myself. So we'll see if I fit the format. But ultimately, I will show everything we hope to do and the way we're doing it and what we have accomplished so far and how we fit into the broader umbrella of this BD-HPP project. So the Metaproteomics initiative was created in 2021 under the UPA umbrella at first. We're now also supported by UPO. We're collaborating with Elixir. And it was in 2021 that we officialized it, and we now cover around 340 plus members, 96 labs in 24 countries. That was from the last update we did early this year at the last symposium in Norway. so it should be pretty up to date. We work on metaproteomics, so we work on the protein of the entire environment, often with the microbes, but also with the host and everything all together. So it's quite complicated. It is at the intersection of a lot of things with other metaomics, obviously a lot of proteomics technique. And so there is a lot of things we can do. And we had a paper already to describe a bit who we are and what we want to achieve. That was when we created the initiative, but broadly, we also have this mission vision, which briefly, we want to share what we can do. We want to interact with people. We want to inform them, and we want to obviously improve the different methodologies, both at the experimental, but also at the bioinformatics, since it's a big part of what we do. So I actually split the presentation following those aspects. First, the communication collaboration among the researcher, education together with microbiome people and proteomics researcher, and the third aspect, acceleration of experimental and bioinformatic methodologies development. So the first one, to increase the communication collaboration. Well, that may not be the most interesting part of the initiative, but it's clearly for us the basics of everything else. I mean, we started, as I said, in 2021, we had some conference before, and we managed to get some people working together. It took some time, but ultimately now it gets way better with all the system we put into place. So we have project now and we can muster, I think the latest one, we have 20 laboratories that took part in the project and we can work with them also. Communication, increasing that communication collaboration was very important. And we use, and we're still using, multiple channels. Some of them are working better than some others. Now, since we have a relatively okay sized community, we can use this newsletter, interact with them. Goes from anything to sharing the latest paper to the job offer or asking for ideas for a new project we can work together. We did try some things like Twitter/X/Blue Sky. Those things kind of like fell out of favor recently, so we're not really using them that much. And also there is a peak of researcher, not really anymore. LinkedIn seems to work pretty well now, but we see ultimately, as I said, what really works for us was actually this symposium. So those symposium predated the initiative. It started at first in 2016 in Germany with, I think, 35 members that went there. We now have it more or less every year and a half, and it's now official and organized by the initiative, so it's working pretty well. So we had them in second 2017, 18, 2021, where actually the Metaproteomics initiative was officialized. And really for us, that was the thing that worked best. And that's also in 2018. Actually, I don't have a picture for that one. Maybe we didn't take one, where we created the first collaborative work among all the researchers that were coming to the symposium. So they were invited to take part in some contest. And that was his first keystone, first work that really helped us putting the initiative together and keep on that momentum. So the latest symposia were in 2023 and 2025, where we gather, I think, 130 people and maybe 85 in Norway. So we're gathering people on a regular basis, and it works pretty well for us. So that's really what we keep on going. And hopefully we'll be able to get more people and more interaction even. The second aspect for us was education of metaproteomics for microbiome and proteomics researcher. And it's not so much educating people as it is for us interacting and collaborating. Obviously, we do organize a workshop, notably at SMS or at UPO, or recently we had at Advance in Proteomics and Technologies in India. We're trying to have a workshop at China UPO, October 2025. So we do have those workshops and those tutorials and training, but very much for us is the interactive part and the collaborating part that is interesting for us. So as I said, metaproteomics is quite at the intersection of many things, with the bioinformatics, with the microbiology, with the other omics and metaomics analysis. So we had actually last year a Loring Center workshop in Leiden, Amsterdam, where although it was organized notably by the Metaproteomics Initiative, the focus there was very much to interact and collaborate with all the other metaomics and try to integrate it and get it better and work better together. That's really what we need to do more and what we want to redo with this initiative. And that works pretty well. I mean, just this year we had a couple of nice papers that came out recently. We had, for example, this microbiologist guide to metaproteomics, so it's extensive review that covers absolutely everything for a microbiologist that would like to get started with metaproteomics, from planning your experiment to the MS acquisition to all the bioinformatics steps that can be more complex in this field and really can be accessible to everybody. From this Lorenz workshop, we actually have also a viewpoint from microbiologists that don't do metaproteomics and actually came back to us and said, okay, that's why we don't do it. That's where we need to, work with each other to collaborate. Those are the things where we believe could be interesting, but we don't know how because we don't know you. So all those things are super interesting for us. And actually, we're also going to have later this year, we have an agreement with Nature Microbiology for a perspective on integrated metaomics, again, from this Lorenz workshop. So that was a very good thing that we did there, trying to meet all together and interacting. But obviously, it's nice to work with others and we want to do it more, but we also have our field that we want to develop, that we want to improve, to push further. And as I mentioned, in 2018, and was published in 2021, we had this first, what we called critical assessment of metaproteome investigation. At that time, CAMP-P. Now we have CAMP-P1, CAMP-P2, CAMP-P3, and planning on the others. So I think on last year's report for REPO, we actually mentioned that we had those CAMP-Ps in the pipe, and soon the result will come out. Now it's pretty cool because they are out. So we have 2 papers with 2 different CAMP-Ps that assess 2 different things that are important for us to keep going forward. So the first one is actually this CAMP-P2, which work on the assessment of sample processing methods, so stabilization of sample. And here I'm just going a bit over the biology and the project we did there, so you see how it fits in the broader part. But for that project, for example, we had stool sample. We also work with environmental sample, but here I focus on the human stool. We had that flash frozen as a control, and that was shared across 8 different labs, in that case all over Europe, I think, with 5 different stabilization methods that were supposed to be tried and replicated by all those different labs. Then those stabilized samples were kept for some time there with everything that can happen in different labs, different places, different variation there. And they were shipped also across the sea, also in the US and some other places in Europe for then MS acquisition, and they were analyzed by different bioinformatic tools. So really, like, if you had to do a collaboration, if you had to sample in one place, send it to some others for some pass and to some others for some analysis, how can we control all of that? And obviously, there are some methods that have their own pros, their own cons. Some are more reproducible than others. Some find closer to the actual truth from the flash frozen, which we consider to be the grand truth here, the control. Some are very much reproducible, some less. And so doing that, we could actually have a look at how good they were performing each method across, also not just somebody that's very much used to that method and expert in that one, but also across people that never heard or never worked with that method. So we could see the variation. Some methods are easier for random lab to implement than some others. And obviously we can look at how good they are doing at finding, well, the number of taxa or the abundance of taxa that were supposed to be present, and also the function and that were supposed to be expressed that we could find in the flash frozen control sample and the others. So with all that, we can actually rank the different approaches and also give feedback on which one is maybe easily implemented, maybe not the best, but more easily implemented and more reproducible. Some maybe a bit harder work, but way better to find the exact ground truth. So it's a stepping stone also for us, because we can say to the community, here it is. That are the options we have. We have tried them all across all the lab, and here's the rank depending on the condition and what you want to get out of it. So a very good stepping stone for us and the community. Then we had another project, in that case, fully on the bioinformatic part. So in that case, we want to assess the impact of bioinformatic decision. So we created a data set where we put peptide of microbes in there. That's the list here. Some of them were very closely related. Some of them were relatively distant from each other. And we give them the raw files. There were different combinations of abundance, different mixes we made. And we also give them a database. In some cases, I mean, with taxa that were present in the sample, and sometimes with taxa that were not present in the sample. And we, on purpose, made it very hard for the community. So we give them those raw files and this FASTA file, just do a search, find us the taxonomic profile of this community. You can do whatever you want. But we made it extra hard for you because we wanted to highlight the different weakness that we could have and how we could fix it, and if some methods were more robust to some trap than some others. And here you can actually see, well, already two groups. One is very diffuse, one is very far away from the ground truth, which is here. Well, they chose to use a different database, like usually a general catalog that was not fitting at all to the sample. So obviously it was way harder for them. But even to the one that is actually close to the ground truth, you can see some distribution. And that's where we are now trying to figure out which method works best with which parameter, which decision or post-processing help them to best resolve the trap we put them. So some fell more easily in some of the traps, some not, and vice versa. So we now assessing that and feeding it back to the community. So now the plan is actually to give them recommendation with what we've learned, let them do the analysis again and see if they can get better, and then bring it for recommendation. Obviously, this thing is a very simplified community. We also work, and sorry for the picture, it's a bit simplistic, but that's where we are now with that project. It's a bit more complicated. We also give them a human gut microbiome in 2 different conditions, and fevers disease. And in that case, we don't have a ground truth, but for that particular project, we have the entire multi-omic picture, so we have a good idea of what's going on. And we know that there are some taxa that may be harder to find at proteomics level, so we want to see if those persons can actually find them and link it to the disease actually. We don't have the ground truth, so we cannot make the exact same stat and really rank them, but at least we can see cluster, we can see where they find. And if we had to put some kind of a ground truth, that would be here, so you can see at some submission actually very close to what we expect to find. And obviously it's way harder, this representation, where to just show you the synthetic community that was easy. There was 16 taxa in different abundance, or sometimes even the same abundance. When we work with microbial communities in human gut, that's way harder. But that's where we are trying to see really how best some method works and how badly, let's say, they fall in some common pitfalls or traps that we can set up for them. So that's another stepping stone, because for us, then we can provide, again, recommendation to the community depending on the complexity, the type of sample, and the question you want to answer. If you're more interested in who is there and what they do, and how abundant they are, the low abundant one, this kind of thing, we can feedback recommendation and particular bioinformatic method that they can use to best answer their question. And finally, a third project that we have right now, it's not really a project, but something that we want to provide also. As we can see with all the contest and comparison we made, there's like a lot of different workflows and methods that people can use. We actually ask people to report what they find, and even with a template we give them, sometimes it's very complicated for us to compare that. Everybody has his way to report that, which makes, well, reproducibility, comparability, and integration quite difficult, and we see that even in our control setup that we have. So we're working now on a metadata in proteomics. And that's also why at the beginning we said we really want to interact with people, because we don't have to reinvent the wheel. There is this SDRF proteomics that already exists. This thing captured all its template that we need for all the MS analysis. But then there is also this MLXS for this minimum information about sequence framework that can capture all the information about the environment, also microgenomics or metagenomics. So for us, we can actually implement that part as an extension to this SDRF proteomics, to have this SDRF metaproteomics. And that's going to be another stepping stone for us to really improve how metaproteomics work and how we share our result and how we connect. And we have more projects like that in the pipe, but all are with this main idea that we want to improve the field and standardize it and really facilitate it to then later on have this broader project. So we have in mind, for example, this global healthy metaproteome, for example, where we want to look at the gut of the human and the whole global. It's not for this kind of project where we start deciding what kind of analysis we're going to do, what kind of standardization method we're going to use, what kind of stabilization method we're going to use. So we have all those pre-projects before we can actually set up all those stepping stones and then actually have this broader, bigger, at the global scale project we can have. So that's where I think we fit in this context of this BD-HPP project, with like building all those things here. We can then later on participate in those bigger projects. So that's it. That's for the presentation of this Metaproteomics initiative. There is a lot of people involved. We have this, that's the board, the current board, but we also work as with an open extended board policy. So all our meetings are actually public. If you're just a member, you can join us and see what we're doing and actually help or participate, because right now we gather people, it's cool, but it's always very often the same 20, 30, 50 people. We always try to reach out and get more people involved and even leading those projects. We will succeed as an initiative. At some point I can just leave, and I know that we'll keep on rolling and probably will keep on going every time new people. So that's the idea. So don't hesitate to join. And there is obviously, as we mentioned at the previous presentation, collaboration across the different initiatives. So that will be also pretty cool to discuss all that. Thanks.
Okay. Thank you very much, Bernard. It was very interesting for me to hear and see what the projects look like, especially the last project that, fecal proteomics that you sent to different labs. I think there's a lot of, I don't see any other questions. That's why, if you're okay, I could ask a couple myself to start and see if anybody else would answer a question. The two questions about the experiment that you've done between different labs. How big is the database was that you provided to people? Was like very close to, set of area that you would like to see, or it was very broad database that you provided in the first round?
So there were 16 taxa present, and you give them a database of 25 taxa.
Okay.
And that database was built also to make it hard. So some of those false positive, there were false positive, obviously, to start with. And then some of them were close related to what was actually present in the sample, and some of them were quite distant. So we can also assess how precise people are, because, I mean, false positive, if you find the exact same species but not the same strain, that's less bad for us. Let's say that if you find completely something that should not be there at all and not closely related. But people could choose to use their own database or your own catalog or UniProt or like unspecific reference if they wanted to. And actually, that's what we see in the very far distant part of the plot. That's where people had this, they analyze this very small community with these very broad databases. But that's also why we have this biological dataset, because it's not fair maybe to compare those tools that use broad database on well, synthetic community that are almost never found.
Mm-hmm.
So we have also this, well, human gut metaproteome, and there they could either use the database, or we provided them a sample-specific integrated metaomic database, so metagenomic and metatranscriptomic, that was co-assembled to give a set of open reading frame. So they could have a specific database still if they wanted.
Okay, yeah, thanks for clarification. And another one about this human gut proteome project that metaproteome project that you have in mind. How physically you envision this? Is it going to be again similar to the fecal proteome, that you would have a certain sample that you can redistribute between lab for them to analyze, or the people will basically try opposite? They will just do analysis of their own samples that they have and then provide data to give you more different type of proteomes rather than single, proteome that have been redistributed.
I mean, we have to define all that, but it's most likely going to be the second option. So the whole idea of this campus that we have right now is to define the set of tools and approach and standardize that, and then we can provide that package, and then everybody across the different lab, the different places can analyze their own samples. So we can start to gather all this information and then have this broader picture view, and those samples will be better comparable because they will be acquired and analyzed with this standardized method that was first assessed to be the best for that type of sample. So first, it's very much like a progression. So we first establish what we need and how we need it, and then we can release that and have this broader project.
Okay. Yeah, and then just a suggestion, I think this is great initiative, and it's definitely would be in the very much interest of the biology of disease area, because clearly establishing a normal healthy gut versus disease perturbed gut proteome is one of the biggest subject that currently is undergoing in many companies. That's why I think there is a suggestion will be, and maybe we should just talk offline about this, that how we can actually involve the pharma company into this journey, because clearly all the pharma company do have such project either already going or in plans to do so. And so if we could just somehow make this community larger to involve pharma into this area where they could provide the analysis of, say, unperturbed type of analysis, that there is not much anything related to drug or any treatments yet being involved, but they could actually facilitate that area where there are, first of all, different countries, which is important, would be contributing to see the heterogeneity of such proteome across the world, as well as, clearly you could just see what the pharma companies is doing, and then for themselves, it would be interesting to compare their own studies versus what you can in the end provide as a larger cohort study.
Yeah, exactly. And that's also why we needed to have all this established method, because then we can really make it shareable and comparable across all those different labs and different samples. So yeah, cool, thanks.
Glad to hear, and I'm happy to work with you on try to establish and reaching out the larger community for this type of project. Thank you, Benoit, for your time and presentation. The next speaker would be Claudia Torteca. She's going to present a next initiative, which is a single-cell proteomics initiative, which I'm part of as well. And I'm very much excited to see the progress of this young initiative. And we will hear from Claudia where we're going with it. Thank you.
Thank you, Bogdan. Can you guys see my slides? Awesome, very nice. So I think that's a very nice trajectory of very established initiatives to a relatively new one to us, who was established 2 years ago, basically. So we're very, very early in our development, and I'm basically gonna introduce the team and what we're gonna be up to, and how everyone who's interested can be getting involved with this. So as Bogdan was saying, we're part of the BD-HPP single cell initiative, and the reason why we're actually working together on this, if I could advance my slide. Actually show you. Well, here we are. So the reason why we're actually doing this is because we really want to focus on biological heterogeneity. And we all know that heterogeneity in the human body and basically in every single organism is imprinted in multiple different levels. We're not just talking about interpatient variability, where we all know every single patient is very unique. But we're also talking about very different origins of disease or developmental differences. So how this has been studied in the past or very early on was mostly using microscopy. Then it was expanded, obviously, using single-cell sequencing approaches. And they really, really nicely contributed to the understanding of biological processes on multiple different levels that are really, really important to understand these biological processes. And more recently, in the last couple of years, mass spectrometry has started to contribute to understanding this heterogeneity. And we have started to see a lot of technical developments in that area, really starting from very early on. Bogdan and Nikolai Savov presenting scope to whatever everyone is trying to do right now. Really, a lot more people are getting involved, and we're really excited to see the developments there and to interpret and to be really getting new insights into disease and developmental progression. The main aims of our initiatives has multiple different levels. And number one would definitely be providing guidance for new users. The team has been struggling for years. We have all gained a lot of experience in what to do and what not to do, or in what way you could overcome some of the challenges. And I think it is for us really important to share this knowledge with the community and make it easier for new users or even established users to go a different route. And that comes alongside with establishing the foundation of quality controls, which is for us extremely important that everyone adheres to specific quality standards. Because we're dealing with single cells, we're dealing with really, really low sample inputs, and therefore we're challenged with a lot of diversity from technical problems or losses throughout the sample preparation and acquisition. So we're really focusing on establishing guidelines or suggesting appropriate quality controls for everyone to use. We're also interested in providing baseline criteria for statistical conclusions. So what we're aiming at is, now that we can increase the throughput, we can have a lot of measurements at a very good sensitivity. We're trying to find out where is a good balance, depending on your biological questions. How many cells should you measure? How well should be your depth of analysis? What is an acceptable variability between your samples? And that actually feeds everything together into a good scientific practice for single cell mass spectrometry data, where we really want to come up with almost a little bit like guidelines that we want to provide to specific proteomics journals, and hopefully in the future expand it beyond just the proteomics journals, to really require very in-depth reporting on those very noisy data sets. And we want to present and compare a lot of sample preparation workflows, since everyone in our team has their best practice, and we really want to showcase what we can do and what would maybe be the most appropriate workflow for you and your biological question. And then we want to compile different instrumentation. So we want to, as best as possible, compare different instrumentation setups, and depending on whatever you have in your lab, to guide what could be your expectations from your single cell analysis. And then lastly, obviously, we also want to recommend analytical strategies in the terms of what would be the best appropriate analysis parameters, how to make QCs post your acquisition, and provide solutions for robust data interpretation, or even guide users of which software packages are out there and how you can explore your data and make some meaning out of everything that you have generated. And when I was preparing the slide, I was like, our team is expanding, and I'm very, very happy to work alongside all of those amazing people. And I thought that we're actually generating some traction and that we're expanding and expanding. And then I saw everyone else and how many people they have on their teams. And I was like, okay, well, it does take a village. We're a small village right now, but we're getting there. So that's basically us right now. I have the pleasure of chairing this amazing team together with Irvin Shu from the DTU. Our initiative was founded by Bogdan 2 years ago. Our newest member, Tina, specializes on bioinformatics. And then we have Zen, who's obviously contributing extremely in-depth knowledge. We have Ryan, just such a great figure in the field, Carl, and obviously Akash. All of them are amazingly contributing knowledge, instrumentation, and just a lot of experience. But we really want to increase our team, and I will go into detail a little bit about that later. Now, what we wanted to do, what we started out with basically establishing the team. What can we do? How can we do it? And how can we improve the understanding of inter-laboratory studies when we generate one sample, like one sample preparation was frozen down and sent out to different labs. And what I'm trying to illustrate here is the essence of our first single cell initiative poster that we presented at International HPP last year in Dresden, where we started to compare what if the same sample is being prepared, centralized, and then being sent out to everyone. And what we included was single cells that were treated with a vehicle and single cells that were treated with a drug. And we really wanted to see what can we get out of this in terms of identifications, fold changes, what is getting a little bit of a feeling how different these analyses would be. And we ended up having 6 different setups with diverse instrumentations in terms of LC and also mass spectrometry. And then all of our data got recompiled and we performed centralized data analysis, because what we think is really important is that regardless of what data analysis workflow you choose, if you try to compare different samples, you need to follow the same exact data analysis strategy. And while we think that is obvious to for us, it is not always the case. So we really made sure that all of this is basically aligned and following specific requirements that we laid on ourselves. And this is what we found. Just a very, very brief overview of the different setups. We're looking at 20 cells, which is obviously a little bit of a higher input than usually we're dealing with on a single cell level. We're also looking at blanks, which for us is extremely important because we're so close to the noise level. We always want to measure blanks alongside. So we see what our background is on the instrument, respectively. And then we have our 2 different cell treatments. And what we could see is actually that along all of these setups, we have 6 different setups with a lot of different combinations. Overall, the identifications are very, very comparable. And that was very encouraging to us for us to see that we can actually generate a very comprehensive data set, sample set, freeze them down and send them out to all of our different labs and acquire and get pretty much comparable data. However, we've found a lot of flaws in our initial setup, which we basically now use to see what we can generate, what is important to us to really consider for our big studies that are important for us. And I want to introduce those studies that we're anticipating. Number 1 would be our interlaboratory study, where we're really focusing on what can we learn from single cell data and how stable is our data sets. And for this, we are going to be collaborating, and we're going to be providing outsource sample preparation to really have unbiased samples. Then we're going to use identical sample acquisition parameters, just different instrumentation in different labs. And then we're going to be focusing on diverse and comprehensive quality controls. We're going to be providing central data analysis, and then we want to share all of this with the community. And in our secondary approach, we're going to be working with comparisons of optimal workflows. So whoever thinks who has the best combination of sample preparation and acquisition strategies, we all go for that, and then we find out who's actually right. And to go a little bit into detail, why we actually want to outsource sample preparation. And we're very glad that we can be working together here with Selenium, where Anjali is going to be helping us with providing the samples and shipping them out to the individual labs. And what we're trying to anticipate here is really to minimize the cellular variability right from the get-go, and then go through the entire sample preparation workflow using the exact same chemicals, the same robot, centralized handling, and then shipping out the samples to everyone in the initiative. And then we are going to be acquiring with our setups in the individual labs. We're going to be using commercially available consumables in order to really have reproducible setups within the different labs. And we're going to use, obviously, state-of-the-art HPLC and mass spectrometry instrumentation. We're going to be providing a couple of different instrumentation setups, but very, very comparable. And we're going to be using standardized gradients and acquisition methods. Now, when we're looking at the comprehensive quality control samples, as I was mentioning, we really are focusing on diverse quality control setups to model and really compensate for distinct variabilities that are not biologically derived. So we want to look at qualitative and quantitative quality controls. Did our sample preparation work? Did our shipping work? Was the thawing successful? Was the injection successful? We also want to provide negative controls because we really want to make sure that we're measuring whatever we're injecting, what we're interested in, and not just the carryover from 5 people that measured before us. And then we want to include sample preparation site-specific samples. So really, whenever the shipment has been done, we want everyone to include their own QC values to see if the instrument has been performing well during the acquisition. Then we're going to be performing centralized data analysis, and this is going to be majorly driven and really nicely supported already by Tina. She's helping us with coming up with good scientific practice on a more statistical level, what is really necessary to provide good quality data and to demonstrate good data reproducibility. We're going to be working on qualitative software tool comparisons, and most important, really provide a platform that can be used by multiple different users to evaluate your quality of your data set and look into the optimal usage or the optimal tool that you want to run your single cell analysis in. And lastly, as I was saying, we want to summarize our finding and provide public access, not just to the data sets, but to everything that we found throughout this process. What we consider extremely important for the sample preparation, the acquisition, and what is important to make valid biological conclusions. We want to obviously grant access to the raw data so everyone can explore and see what we're up to and what would be an expected overview of your raw data. And we're really looking forward to incorporate a lot of the community feedback. So what the team and I have been doing is walking around on all different types of proteomics conferences, trying to talk to as many people as possible. And gather new input, getting new feedback, and really broadening our team to see what we can all learn from each other. And I think that's the most important aspect. Like, again, what we can learn from each other and how can we help each other to make this very, very young field be more reproducible and really convincing to biologists to perform single cell analysis. And that's it.
Thank you very much, Claudia. Very nice representation of the initiative and what we try to accomplish and do. I think we all could make sure that we share such initiative between broader community. And exactly this today, actually presentation was about that we would like to share all those different initiatives that was presented today and actually represent what each initiative can do and somehow just connect the outputs of the initiatives to other fields. For example, what we try to do in the single cell proteomics might be very well served to either to the metaproteomics at some point, or maybe to different organisms. That is actually very important to see what the single heterogeneity is. And as well as we all probably aware of that RNA-seq was doing single cell for quite some time right now, and we have a gigantic amount of the data that's already been acquired in the field. Now, I think if we just could add an extra layer of knowledge about the proteomics heterogeneity, that could actually help us quite a bit to see what extra area where we cannot see at the bulk level could be open for us to do another analysis. And so I thank you very much, Claudia. Thank you, Benoit, and then Suzanne for representing today your each initiative. They're all very exciting to me and very close to my heart to see how we could bring this new knowledge towards understanding how the biology of human beings are working, as well as actually how we can contribute to the health of the planet as well. And altogether, I think we can actually move this initiative towards the larger area of the HUPR, try to understand the proteome and try to understand which proteins actually functions are. I think all of us will contribute. They are very much specific area, but in the same time, I think this particular presentation today very well show that we actually do one big project all together. Just doing this from the different angles and different capabilities. But for example, what we learn from the very noisy data and heterogeneity in our own field, I'm sure will be much more important for Benoit, for your guys' analysis, because that's what you're dealing with as well. Very closely related species in the gut of the human beings that are not very abundant to deal with. That's why all those, initiative and sharing knowledge, how to do and what to do, especially with the new instrumentation, which provide us with absolutely amazing capabilities lately, we could just help each other to do so. That's why I would like to thank you very much for your presentation today. And would like to make sure that everyone who is listening to us today and seeing this video, please share your knowledge. Please ask your questions. You've been seeing that there is emails being provided in the end of each initiative. If you would like to join initiative, please do not hesitate to ask about joining either initiative or propose the project if you do have projects that are related to each initiative that you've heard today. Okay, well, with that, again, thank you very much, and looking forward to see everyone on the HUPR conference.
Thank you.
Thank you.
Thank you. See you.
Bye.